Good afternoon, everybody. In the lab today, we are going to talk about different molecular methods that can be used to characterize genotypically a bacteria. So before we start talking about some methods used for genotypic characterization of microorganisms, let's define some general concepts. So, DNA. DNA is a long molecule that contains our unique genetic code. So it holds the instructions for making all the proteins in our body. Then RNA. RNA is a nucleic acid similar in structure and properties to DNA, but it only has a single strand of bases, and instead of the base thymine, RNA has a base called uracil. The transcription. Transcription is the first step during protein synthesis when the DNA is a gene in copy to produce an RNA transcript called messenger RNA. Then translation. So translation is the second step during protein synthesis where the message from DNA for making a protein has been taken to the ribosome and a protein is constructed with the help of the tRNA. So now let's talk about the central dogma. So the central dogma is the process where the DNA is converted into a protein. A protein is also known as a functional product. So this involves the DNA instructions being converted to an RNA message, which is then translated to produce a protein, a string of amino acids. So DNA replication is the first stage of the central dogma, and it is the base of all molecular processes. So several techniques, molecular techniques, have been developed and extensively used for typing foodborne pathogens. And what is a molecular typing? A molecular typing is a way of identifying specific strains of microorganisms, such as bacteria or viruses, by looking at their genetic material. It is mainly used to pinpoint the source of foodborne outbreaks. However, it can also be used to identify which microorganisms are the most virulent and cause serious diseases, the ones that are resistant to antibiotics, and those microorganisms that are able to survive and multiply. So last week we talked about different methods for phenotypic characterization. Today we are going to talk about some molecular methods used for molecular typing or genotypic characterization. So first we are going to talk about the polymerase chain reaction, also known as a PCR. This is a technique used to amplify or copy small segments of DNA. So in order to amplify a segment of DNA using PCR, first the sample is heated, so the DNA then adheres or separates into two pieces of a single strand of DNA. This step is called denaturation. Next, an enzyme called Taq polymerase, that is a heat-resistant enzyme commonly used for PCR amplification, synthesize, builds two new strands of DNA using the original strands as templates. This process results in the duplication of the original DNA, with each of the new molecules containing one old and one new strand of DNA. This step is called the annealing step. Then each of these strands can be used to create two new copies, and so on and so on. This last step is called elongation. The cycle of denaturing and synthesizing new DNA is repeated as many as 30 or 40 times, leading to more than 1 billion exact copies of the original DNA segment. Some examples of traditional polymerase chain reaction or PCR methods include the multi-locus BNTR analysis, also known as the MLBA, and the multilocus sequencing typing, also known as the MLST. The MLBA is an abbreviation of the multi-locus variable number of tandem repeat analysis. This is another technique used by microbiologists to generate a DNA fingerprint or a bacterial isolate. Microbiologists usually perform MLBA after they have performed the pulse field gel electrophoresis, the PFG, that is a method that I'm going to explain later, so they can get more detail about the type of bacteria that may be causing an outbreak. So then we have the multi-locus sequencing typing, also known as the MLST. This is a typing method that combines the sequencing of several housekeeping genes. Remember, we already talked about what is a molecular typing. So the MLST has been designed to analyze and compare genetic variation in worldwide collection of bacterial pathogens. It gives important information about the nucleotide divergence of the core genome, the clonal origin of one group of strains, the recombination rate, and the phylogenetic relationship among strains. So PCR technology was limited to qualitative and or semi-quantitative analysis due to limitation on the ability to quantitate nucleic acids. At that time, to verify if the target gene was amplified successfully, the DNA product was separated by size via agarose gel electrophoresis. Ethidium bromide, a molecule that fluoresces when bound to dsDNA, could give a rough estimate of DNA amount by roughly comparing the brightness of separate bands, but was not sensitive enough for rigorous quantitative analysis. Improvements in fluorophore developed and instrumentation led to thermocyclers that no longer require measurement of only end product DNA. This process is known as a real-time PCR or quantitative PCR or qPCR. The key feature in real-time PCR is the amplification of DNA is detected in real time as PCR is in progress by the use of fluorescent reporter. The fluorescent reporter signal strength is directly proportional to the number of amplified DNA molecules. So there are many techniques used in order to perform RT-PCR or qPCR. Some of they are the back system, the GeneDisk, the Microsec, the Assurance GDS, the RAP-AD, and IQ from Biorad. First, we have the BACS system. So the BACS system is an instrument that features two types of polymerase chain reaction detection technologies. So the real-time and the endpoint. The endpoint is when all the cycles of PCR are completed. Also, it's composed for table reagents and optimized media to detect unwanted bacteria in raw ingredients, finished products, and environmental samples. The BACS system provides clear yes or no results and it is easy to read amplification profile with no need for subjective or expert interpretation. Then we have the GeneDisk system that is composed by the GeneDisk plate and a GeneDisk cycler. The GeneDisk cycler is a robust real-time PCR platform for rapid, sensitive, and specific detection of microorganisms in a matter of hours. It is comprised of a base unit plus up to 7 subunits. Each of the units can be hold one GeneDisk plate. The plate's unique proprietary design enables the detection of a range of microorganisms simultaneously within the same sample DNA extract. The cycler performs gene amplification in the plate, and each plate can be used to test either 6, 9, or 12 samples in parallel. When all subunits are in use, the cycler can analyze up to 96 samples in 1 hour. Then we have the Microsec. The Microsec is the microbial identification system that is a method that tests eubacteria, yeast, and filamentous fungi. It rapidly identifies slow growers and non-fermenters in less than 24 hours that are often impossible to identify by conventional systems. No prior knowledge of the organisms is required since the system uses universal domain level primers. The Microsec system eliminates subjective, phenotypic, or growth-based test interpretation since it is based on RNA gene sequences. The results are reproducible across laboratory and over time, and because the system employs the PCR process, culture conditions do not influence results. Then we have the Assurance GDS. That is a method that uses proprietary magnetic particles to capture the target organisms from the enriched sample. This method collects and transfers the concentrate target to 8 samples at a time. The Assurance GDS utilizes highly conserved target gene sequences that ensures greater specificity with fewer indeterminate or false positive reactions. Also, multiplex DNA amplification, which allows simultaneously detection of multiple targets within each amplification tube. So there are other methods used to identify microorganisms genotypically without the need of using a PCR step. One example is the pulse field gel electrophoresis, also known as the PFGE. This is a laboratory technique used by scientists to produce a DNA fingerprint for a bacterial isolate. This is a powerful genotyping technique used for the separation of large DNA molecules, the entire genomic DNA, after digesting it with unique restriction enzymes and applying to a gel matrix under the electric field that periodically changes direction. The PFGE is a variation of an agarose gel electrophoresis that permits analysis of bacterial DNA fragments over an order of magnitude larger than that with conventional restriction enzyme analysis. It provides a good representation of the entire bacterial chromosome in a single gel with a highly reproducible restriction profile, providing clearly distinctive and well-resolved DNA fragments. In this slide, you can see on the picture on the left the process of the pulse field gel electrophoresis since your bacterial culture up to the final result. On the picture of the left, that's how it should look in real life, the results from this method. Also, we have the whole genome sequencing. That is a method that determines the order of bases in the genome of our organisms in one process. The whole genome sequencing is conducted by following these 4 main steps. First, the DNA sharing. So the DNA is cut into pieces that are small enough for the sequencing machine to read. The pieces are composed of millions of bases: adenine, cytosine, thymine, and guanine. Then the DNA barcoding. So the scientists add small pieces of DNA tags or barcodes to identify which piece of shared DNA belongs to which bacteria. Third, we have the whole genome sequencing. So the barcoded DNA from multiple bacteria are combined and put in the whole genome sequencer. The sequencer identifies the adenine, the cytosine, the thymine, and the guanine are bases that make up each bacterial sequence. The sequencer uses the barcode to keep track of which bases belong to which bacteria. Then we have the data analysis. So the computer analysis tools are used to compare bacterial sequences and identify differences. The number of differences can tell you how closely related the bacteria are and how likely it is that they are part of the same outbreak. So the whole genome sequencing provides more detailed and precise data for identifying outbreaks than pulse field gel electrophoresis. Also, it's a method that its price really high. On the picture on the slide, you can see the whole process step by step that involves performing a whole genome sequencing.